Let me start with my favorite biologist, a quote from Darwin. Our classification will come to be, as far as they can be so made, genealogies. And will then truly give what we may be called the plan of creation. So Darwin had a dream, and that is that one day we would be able to classify organisms on the basis of their evolutionary history. And of course, now we are able to do that. The question is, to what extent? So to situate the genus Metschnikowia, for those of you who are not familiar, within the yeast, so this is a tree taken from Kurtzman and Robnett, 2013, showing the Saccharomycotina, and you can see at one end the well-known yeast Saccharomyces cerevisiae, and at the other end the not-so-well-known Metschnikoviaceae, two genera. And you can see already by the choice of taxa that the author of the tree has already decided that the genus Metschnikovia should be subdivided into two genera. It's almost a losing battle, and it's been going on for a long, long time. The genus Metschnikovia was named from a species, Monospora, that was actually described by Metschnikov himself. Eli Metschnikov, one of the founders of immunology. And the battle has been raging ever since. People have been trying to change the name Metschnikovia. There have been proposals for Anthomyces, Monosporella, Metschnikoviella, Chlamydozyma, and it goes on and on. One proposal that has actually survived for quite a while is the advent of the name Candida for species that did not have a sexual cycle. But we're about to put an end to that due to the one fungus, one name rule. So Candida is about to come to an end, at least within the genus Metschnikovia. As I said, it is a battle. The forces of evil are still there trying to split the genus into all kinds of smaller genera. However, I think the force will prevail. All right, everybody's been showing you pretty pictures of beautiful macrofungi. It's more difficult to do that with Metschnikovia because it's largely unicellular. However, as far as yeasts go, the morphological diversity in Metschnikovia is quite remarkable. The one unifying theme is that the ascospores, which are a meiotic product, are actually needle-shaped. Two ascospores form per ascus, and they are formed during meiosis I. And what happened during meiosis II is still a mystery, and I'm hoping to solve that mystery eventually. In terms of habitats, Metschnikovia has many different habitats, but there are some recurring themes. The largest clade on top is almost exclusively from small nitidulid beetles that occur in ephemeral flowers, as mentioned this morning by Meredith. A number of species are well known to inhabit nectars of many plants, and so I show a milkweed as an example, the second photograph. A whole subclade is associated with aquatic invertebrates, and quite a large subclade is associated with the surface of fruit, and then there are all kinds of other sources. So if we consider the ascospores a defining characteristic of the genus, the other defining characteristic is that there's very little diversity at the level of physiological phenotypes. So let's not talk too much about that. So the new code says only one correct name for a fungus species and forces us to re-examine the concept of genus. So part of my task will be to reassign some Candida species to Metschnikovia and to make sure that generic assignments are correct. So let's look at an example of where things are going to be easy. This is part of a tree that contains a particular Metschnikovia clade. It doesn't really matter which, but this is an example where old Candida species are well embedded into a fairly robust phylogeny, so renaming those can be done without much problem. At the other end of the Metschnikoviaceae tree, is a bunch of Candida species and a few Clavispora species, a sister species to Metschnikowia, for which the phylogeny is not very reliable. So all the internodes that have arrows pointing at them are poorly supported. And so we need a better phylogeny before we start messing around. The genus Clavispora itself, which is circumscribed in a box on this illustration, as you can see, is, well, you'll have to decide whether that's polyphyletic or paraphyletic. I had a question about that on my exam, and most students got it wrong. Maybe it was me who got it wrong. So I go from the assumption that there is a true phylogeny, and that our task is to figure out what it is. This here is not the correct phylogeny. It's the phylogeny that I used in the last monograph. It's based on D1, D2, which is almost impossible to align correctly for that group of species. Here's a more recent tree by Guzman et al. 2013, based on 4 loci, and it looks much nicer, but it isn't. It is not reliable at all. So let's explore part of this tree and see how we can get to better phylogenies. I will focus on my favorite group of yeast, the so-called large-spored clade, as I said before, associated with nitidulid beetles. It makes beautiful asci with very interesting morphological features. From that clade, I will extract a small subclade of species that are exclusively Hawaiian in their distribution. And so based on D1, D2, if we examine the details of the topology of the tree, we find Hawaiiensis and Hamakuensis are sisters, and Maui Nui Ana is a sister to that pair. If we go and examine Guzman's tree, we got a different story. We find that Hawaiiensis and Mauiensis are sisters, and Hamakuensis is basal to them. So, contradictions. Unpublished data from my lab, we looked at 7 anonymous polymorphic loci, and we get yet another topology, topology C, where Hamakuensis and Maui Nui Ana are sisters. And so, which is correct? All 3 cannot be correct. It'd be nice to have perfect data all the time. By that I mean strong tree structure with long internodes, equally well distributed. That's not usually what we get. Perfect data are hard to find. Most of the time we get either this, one extreme where there's very little variation between closely related species, or an opposite effect. So let's explore this a little bit. Incomplete lineage sorting is the problem. And there's a very nice example of an analysis of such phenomena in the Great Apes paper by Ebersberger that came out quite a few years ago now, 8 years ago, where he and the team actually took about 25,000 bundles of aligned orthologs and derived every possible tree. So there were 15 possible trees with that group of taxa, and they found that 60% of the trees gave the well-known phylogeny that puts humans and chimps as sisters. But a fairly large fraction of the trees also supported the idea that chimp and gorilla are sisters, and that's the green representation. And that was attributed by the authors to incomplete lineage sorting. And another minority topologies supported human and chimp together, but gorilla and orangutan together. When you have a tree with apes, it's so tempting to put pictures of colleagues next to the tree, so I could not resist. So consider it done. The order is interchangeable, depends on the day. So nothing personal, turn. Next time you'll be human. So the other problem we encounter, and I have not dealt with that one yet, is when we have very long branches and very short internal internodes due to saturation. So this is an example of the effect of saturation. the p distance that we measure is a bad representation of the true d distance. So one dogma is that if you have good data, it doesn't matter what method you use to construct a tree, you're going to get the correct phylogeny. And the opposite of that is that if you have bad data, but you use a very sophisticated method, you get the correct phylogeny. Of course, this is completely hogwash. The central theorem of phylogenetics is trash in, trash out, regardless of the method. And that's my principle. So the solution, find better data. And we have access to better data using draft genomes, whole genome sequences. If you're not familiar with this paper by Lachance and Lachance, I recommend that you find it. 2013 in Nature, where they examine what is the source of problem in phylogenetic data. But one of their recommendations is we must carefully examine the signal present in individual trees and their genes. So we did that. Going through my methods quickly, we purified DNA, sent it to Genome Quebec, where they provided us with Illumina runs, and we were successful in getting good runs with between 9 and 12 different genomes per lane. And I have the privilege of having a colleague who knows how to do the primary assemblies, Tom Siang, so that's been wonderful, and a student who's very good at doing the interpretation at the genetic level. So we sorted almost 1,000 orthologs out of all the genes available. These had good properties, they were sufficiently long, they were sufficiently informative, and we examined all that, so which topology is correct, and we found 3 topologies distributed approximately 60% and 20-20, just as the people had found in the apes, which is interesting. A very regrettable thing for me is that the best supported topology was the one that I didn't like, and so maybe there's something wrong with it. However, I will follow the data where the data point, and I will accept the 63% topology as the correct one. So why are these topologies different, and why should we trust the majority? We examined the strength of the phylogenetic signal and found that the topology C at the bottom is the one that had the best bootstrap support, contrasted to the others. So one more piece of evidence that we must accept that. We also did MCMC trees, and again we found that the posterior probability distribution was the best for topology C. We also asked the question that perhaps the 3 different topologies are due to different selection regimes. So we started examining that question by taking what's between the genes, the spacer sequences that are supposed to evolve in a neutral fashion. We found the same pattern, approximately 20% support for A, B, and 60% support for C. So that seems to be the trend. We also wondered whether there's horizontal gene transfers, introgression, that sort of thing. And to test that, we took the flanking regions of genes that gave the various topologies, A and B on the left, and the resulting phylogenies, again, were in favor of topology C. So from that, we conclude that the history of these species is that shown by topology C. So general conclusion, topology C is the correct one. What do we do in the future? Well, I have a total of 55 now of those complete genomes, so we hope to spend the summer expanding our results to all 55, and eventually to expand that to all the Metschnikowia ACE. And once we've done all that, see if we need to reassess genome structure. Now, I'll again conclude with a quote from Darwin. I will let you read it because it's too many tongue twisters in it. So Darwin yielded a mixture of things that turned out to be right and others that turned out to be wrong. Whether classification is ever going to be simple, I doubt very much. Thank you for your attention.